I'd like to introduce our speaker, give you a brief summary of what her talk is about, and let you know about 3 STEM lectures during the spring semester. This is the last of the fall semester lectures. So our speaker is Dr. AnnMarie Torres. She is currently with the Department of Cancer Biology at the University of Pennsylvania, where she is a Penn-Port postdoctoral fellow doing research at UPenn. She has a PhD in genetics from Stony Brook University, Stony Brook, New York, and a Bachelor of Science in Microbiology from the University of Massachusetts. She's won so many awards and honors that, thank you, that I am not going to even begin to list them. She has quite a bit of teaching experience, and we're fortunate that this semester she's teaching here at PCCC. She's given dozens of national, international, and local presentations, and her most recent publication this year was in the Journal of Leukocyte Biology. So a very accomplished speaker. A synopsis of what she's going to talk about from now until 12 o'clock. The title of the talk is Fat, Not Just a Villain in Metabolic Health. There are benefits of fat. So for years, fat was considered just a type of connective tissue that served no metabolic function. However, this outlook has greatly changed as research has revealed that fat, or adipose tissue, is actually an important endocrine organ, and Dr. Torres is going to talk about it. Before she begins, upcoming talks this coming spring at 11:10 and 5 o'clock on the 4th Tuesday of February, March, and April. In February, the talk will be What's New in Microbiology by Dr. Robert Saran, one of our full-time faculty. In April, exploring a metabolic aspect of the parasite that causes African sleeping sickness, and that's going to be given by another one of our professors, Megan Hickey. And in April, Environmental Activism Today. April is Environmental Month, and that's going to be presented by Russ Johnson. So some very interesting talks in the spring semester. So without further ado, let me present our speaker, Dr. AnnMarie Torres.
Thanks. Thank you guys for all coming, and I want to thank you all for this opportunity to share with you a bit about my work that I've been doing while at UPenn the last about year and a half. I'm just going to start by just telling you a little bit about myself and how I kind of got to where I am today, the path I chose. So everyone has, I guess, a story, how they ended up in their career. Mine is maybe a little different. I hated science and math in high school, and I was really excited to go to college and not ever have to take science or math ever again because you can pick the classes you take, except I found out that that is not true. When I got to college, I still was required to take 1 science and 1 math class. So I went through this course catalog, because when I was in college, it was not on the internet yet. So you had to actually go through the catalog and read this like tiny, tiny print. So I was like, what science class can I take that will be really easy? And I could just kind of get it out of the way and move on, because I was going to be a political science major and go to law school. So I found this class called The Biology of Cancer and AIDS. I was like, that sounds interesting, so let's do that. And while I took that class, I started taking my political science classes, which turned out to be incredibly boring to me, and I just could not sit through a whole class. Meanwhile, I was taking this class on cancer and AIDS that turned out to be really interesting. And I had a professor who just was so excited and passionate about what he taught that it was just inspiring. I found myself actually going back to my dorm and actually looking up information because I was like, I'm just not getting enough. I need to know more. So at that point, I was like, I should probably rethink my career path here because I can't handle. I took another semester of my political science and just could not get into it. So I decided, without maybe putting enough thought into it, to just declare my major as microbiology. So I was wanting to understand all these interesting microbes and how they cause disease. And so I was very interested in host-pathogen interactions. So I ended up just declaring that major. It was probably one of the hardest things I ever did. It was an extreme struggle because math and science never came naturally to me, so I always had to work what I think is very hard just to study and keep up and do well. I decided to get some undergraduate research experience, so I joined a lab, which was really the point where I was like, oh wow, this is really cool, because this is the stuff that nobody knows about. I'm doing research that nobody has figured out yet, and I just really love being on that cusp of big breakthrough, understanding exactly how something works and being able to share that with the world. So I decided after that I wasn't ready to commit to grad school at that point because science was still quite a challenge. So I actually worked for a year at Cold Spring Harbor Labs in New York, where I did some research in cancer biology and also learned some really great techniques. And then I was pretty confident that I was ready to go to grad school, so ended up going to grad school where I studied micro. And I studied this host pathogen interaction. So in grad school I studied how salmonella affects the immune system. So I studied not the kind of salmonella that gives you food poisoning, but the kind that leads to type 1 fever and is usually very deadly. And what we found through my work was that that salmonella prevents the immune system from responding to the infection by interfering with T cell metabolism. So T cells are like a white blood cell, and salmonella actually creates and produces and secretes this protein that prevents the T cells from doing their jobs, and it does it by interfering with metabolism. Metabolism, what is that? So I decided this is a really interesting field, so I wanted to be able to learn a lot more about how microbes may interfere with the immune system at the metabolic level. So I decided I need to go somewhere and learn a lot about metabolism. So University of Pennsylvania has one of the top metabolism groups in the world, and I was fortunate enough to get a position in one of their labs. And that's kind of where I've ended up today. I am in a cancer biology department, and I don't study cancer, as you're going to see, but I study metabolic effects of fat on whole body homeostasis. So I'm going to share some of that work with you guys today. Okay. All right. So metabolic pathways, probably have learned a lot about them in your classes. They're extremely important in both growth of a cell and biosynthesis. So a lot of times when we talk about metabolic pathways, we kind of divide them into these two main types, anabolic and catabolic. So anabolic is building up, making molecules that the cell needs to live. This could be proteins, it could be parts of the cell wall, ribosomes, any of those major molecules the cell needs to live every day. But then there are circumstances where the cell needs just energy. And in order to generate that energy, it's going to break down some of these molecules and just turn it into energy, and that's called the catabolic process. But not all cells are constantly doing both these activities at any one time. So most cells in your body at any given time are in a quiescent state, which really means they're kind of just resting, just kind of hanging out, not doing too much, just doing exactly what they need to live. So this usually involves just generating energy, also known as ATP. But when a cell realizes it's in a situation where it needs to grow and proliferate, if it's at time, just time to divide, or if it finds itself in a situation where it needs to generate energy to proliferate, it's going to start building up biomass. And these are those lipids, those fats, the proteins, RNA, DNA, all the stuff the cell needs if it needs to grow and divide. And so when we talk about metabolic pathways, many times we're discussing what's going on in a proliferating cell, because these are the most metabolically active. And so that's what, when I'm talking about a metabolic cell, that's what I mean, proliferating or growing cell. And the main driver of most metabolic pathways are carbohydrates. And probably the most important one for a cell is glucose, so it's a sugar. So many of you might have seen this diagram before, and I actually stole it from the textbook that my class uses. I'm teaching a micro class right now, because I thought it was just a really nice depiction of the main metabolic pathways that I'm going to actually talk to you guys about. So really, metabolic pathways are driven by this sugar glucose. And so when the cell takes up glucose, it needs to break it down in order to use it to generate energy or to make all those different macromolecules I just told you about. So upon uptake of this glucose, it's going to get broken down through a process known as glycolysis, generating this metabolite called pyruvic acid. Pyruvic acid can go in two different ways depending on the status of the environment the cell's in. Usually, if it's in, a nutrient-rich environment, it's going to go through respiration. So this pyruvic acid, through another process called decarboxylation, will make acetyl-CoA. And from there, acetyl-CoA can go into our TCA cycle and get fluxed through this pathway to generate energy. And acetyl-CoA is actually the metabolite that I studied. And acetyl-CoA is really an important metabolite because it's really at the center of many, many metabolic pathways. So acetyl-CoA is needed to generate fat, it's needed to generate cholesterol, it's needed to generate for amino acid production, it's involved in ketone bodies, which are a really important metabolite as well. And since it's at this kind of center of so many metabolic pathways, my lab generally actually studies the effects of manipulating acetyl-CoA concentrations in the cell. And so this is kind of our cartoon depiction that our lab uses a lot when we talk about our work. So glucose can get taken up by the cell, broken down, again, like I said, through glycolysis to make this pyruvate or pyruvic acid, which can then enter the mitochondria and be used in the TCA cycle, which can be used to subsequently make more energy. But actually, in this very first step of the TCA cycle, this metabolite known as citrate is made and can actually get exported out of the mitochondria back into the cytoplasm, where an enzyme known as ATP citrate lyase, or ACLY, can convert it into acetyl-CoA. And that acetyl-CoA usually is used either in lipid synthesis, so to make fat, or in acetylation. Acetylation is a post-translational modification that can be put on proteins and has some really important effects on gene expression. And this can all lead to growth of the cell. Now, I want to just be honest with you that even though ACL is the main enzyme in our cell to make acetyl-CoA, there is one other enzyme that can do it. And it's called ACSS2. And it makes acetyl-CoA in a different way than ACL does. Instead of taking this citrate out of the TCA cycle, it actually just can take acetate in the environment and literally paste on some CoA and make acetyl-CoA. So it's a really direct process. This enzyme is not well studied, although it's becoming very popular right now. So exactly when it's most useful is not clear. But at this point in time, we don't believe it to be a primary driver of acetyl-CoA production. However, ACL has been very well studied, especially by my lab, and what we've found now over the last several years is that ACL has been implicated in many cancers. So we know now that there's elevated or increased expression of ACLY in a host of cancers: lung, prostate, bladder, breast, liver, stomach, colon. We recently have discovered that ACL may be playing a very important role in pancreatic cancer, so we have a very big project going on in that. We also study this in glioblastomas as well. We found that ACL expression affects glioblastomas as well. So this is actually a super active area of research in my lab, so that's why I'm in the cancer biology department, because we predominantly study ACL's role in cancer. But as I mentioned, ACL is not just, it's important in a lot of things. So actually, when it first was discovered and studied, it only seemed to have a role in this fatty acid production. And we still don't really completely understand how it regulates that process. So I actually am studying how acetyl-CoA may be regulating this production of fatty acids. And so just to remind you, fatty acids, a free fatty acid is just this long chain of carbons, these carboxyl group at the end. When it combines with a glycerol group, right here, all comes together, that's a triglyceride. So that's the fat that's usually stored in your body. And the cell that actually stores fat in your body is called an adipocyte. So adipocytes are actually, their main job is to take up fat and store it. Adipocytes are kind of a cool looking cell to look at because they're a little unique as compared to other cells. So they actually, this big yellow blob in the middle is a lipid droplet, or the fat, and it grows to be quite large, so it takes up almost the whole cell, and it squishes all the other organelles of the cell off to the side. So you can see here, like the nucleus, the Golgi, the mitochondria, everything's squished off to the side because the lipid droplet just takes up the entire cell. And even though this is a cartoon depiction of it, it's actually quite accurate. So if you get to look at adipocytes under a microscope, this is exactly what you see. These lipid droplets actually are kind of shiny, so they come up really nicely in a microscope. It's really easy to see these fat droplets. But their main, like I said, adipocytes' main job is to take up and store energy in the form of fat. And then in a time when the cell needs energy, break down this fat to make energy. Now, adipose tissue, which is entire groups of adipocytes coming together to make your fat, is actually really important in metabolic homeostasis. So for a long time, fat tissue wasn't really thought to do much of anything except store fat, and then when your body needs to break it down, it gets broken down. Over the years, more and more we're finding that your adipose tissue really regulates a lot of important processes, so especially insulin and glucose sensitivity, which I'll talk about today, your lipid metabolism. More and more we're finding connections between your adipose tissue and your immune response. Angiogenesis, which is the formation of blood vessels. Appetite and energy balance is another big one that we're finding adipose tissue can regulate, as well as blood pressure and nutrient transport. Unfortunately, I really wanted to show you how big an effect adipose tissue can have on your body. And my picture, I have this like Mac to PC issue. So the picture did not translate. I deleted whatever showed up there. But basically, I'm going to try to describe it so you can work with me here. So thanks to genetic engineering, we're able to make mice that express proteins or overexpress genes that were of interest. So we were able to genetically, when I say we, the whole scientific community, I didn't do this, genetically engineer a mouse to make it overweight, because it was found that there's actually a protein that adipose tissue makes called leptin that regulates your weight. So when they talk about a person having a genetic predisposition to being overweight, it's actually true. We found a gene that makes a protein that can regulate this. So if we knock this protein out or take it out of the mouse, genetically engineer a mouse, this mouse is overweight. But if we take this overweight mouse and now re-express this protein, it becomes even more overweight. It's a really funny looking mouse. It's huge. It's just like a big fur ball. But actually that mouse is completely metabolically healthy. So it's a really interesting dichotomy. So you can be overweight yet metabolically healthy. And it's something that we're very actively trying to understand. But basically by re-expressing these important proteins that your fat needs to make, we can really manipulate our metabolic health. So these super overweight mice had completely normal responses to glucose and insulin, so they weren't diabetics. They probably had some trouble moving because they were very large, but metabolically completely healthy. So we know that fat can secrete proteins that play a really important role in our overall metabolic health. I'm really sorry that picture didn't come through, because it's a really good picture. Anyway, so as I think I've conveyed to you, adipose tissue dysfunction, so if it's not working the way it should, it's secreting the proteins at the right time or the right amounts, can lead to a lot of really important pathologies such as diabetes, obesity, cardiovascular disease, inflammation, and now we're finding more and more that obesity leads to cancer. So we're finding this in breast cancer, we're finding this in prostate cancer, and now we're thinking it might also be a potential role in pancreatic cancer as well. So just in case you don't know exactly what obesity and diabetes is, obesity is actually, defined by excessive fat accumulation when they measure your BMI, which is your body mass index. So it's like a ratio of your weight to your height. There's kind of a controversy as to whether that's a really accurate way to measure it, but that's still how they do it in the doctor's offices. And diabetes is a disease in which your blood glucose levels are higher than normal. So this is, I should say, when I talk about diabetes in my talk, I'm referring to type 2 diabetes, which can develop over time, as opposed to type 1, which is usually a complete defect in your beta cells in your pancreas. That's usually a genetic disorder that you're born with. So when I talk about diabetes, I mean type 2 diabetes. And unfortunately, obesity is quite an epidemic. So just in the US alone, in the last 30 years, you can see this progressive increase in obesity across the entire country. And even sadder, obesity not only has doubled in the last 35 years in this country, but it's becoming a bigger problem in childhood. So childhood obesity rates have more than tripled since 1980. And as I said, obesity can contribute to heart disease and stroke, type 2 diabetes. And it's estimated in the next 40 years that 1 in every 3 Americans will have type 2 diabetes. So these are kind of like scary numbers, but the stat that kind of, I've heard this stat many times and I still can't wrap my head around it, but in the last year we have reached the point in this world, as a world, not US, just as a world, where problems associated with obesity has outpaced problems associated with malnutrition. So, we think about, especially in a lot of third world countries, people who don't have food, and there's a lot of issues with malnutrition. Well, we have reached the point where obesity is a bigger problem in this world. I don't know what to make of that, but it's kind of a sad situation. There's no good situation for that to work out, but it's kind of interesting. So basically, we're trying to understand the role this protein, ATP citrate lyase, or ACLY, has in regulating fat production, as well as even degrading fat or taking it apart to make energy. So what we already know coming into my project is that ACL levels are suppressed in adipose tissue when we look at patients or animals on a high-fat diet or who are already obese. So we know when you become obese that this protein gets suppressed. And in mouse models, if we silence ACL or delete it so it can't be expressed, we decrease expression of this glucose transporter called GLUT4. So glucose has to be taken up, as I mentioned earlier, right, for metabolism to take place. So if you can't take up this glucose, you're going to have a severe problem with, well, first of all, after you eat and all this glucose is in your blood, you're not going to be able to take it up, so you're going to eventually develop some sort of, insulin resistance problem. So as I said, it can lead to problems with insulin signaling. And then we also know that when we manipulate ATP citrate lyase, or ACLY, in mouse models, we see effects on the inflammatory response, suggesting we're actually seeing inflammation caused by decreasing the expression of this protein. So my hypothesis at this point was that ACL plays a key role in normal adipocyte function. And if you suppress it, or if it's suppressed in an obese situation, then this will lead to metabolic dysfunction, and likely maybe playing a key role in driving obesity and type 2 diabetes. So as I get into my data, I just want to tell you a little bit more about fat. So there's actually at least 3 distinct types of fat in the human body, as well as other mammals. We usually call them white fat, beige fat, and brown fat. They get their names more from their pictures. So these are histological samples, so fat tissue taken out of mouse and then looked at under the microscope after being stained. And so white fat, you can see here, it looks white, and these big white blobs are actually the fat droplets inside the cell. And as you can see, they're pretty large, and you can't really make out any of the other organelles because they're just completely squished off to the side. Brown fat gets its name because it kind of has this like brownish pink look to it, and that coloring comes from the fact that brown fat has lots and lots of mitochondria. It's used to burn energy. I'm going to tell you a little bit more about brown fat in a minute, but this is kind of like the cartoon version of what's up here. So white fat is just, again, like a big fat white droplet. Brown fat has tons of mitochondria that when you stain them, give this like brownish pink color. And then we have beige fat, which is essentially somewhere in between a white fat and a brown fat depot. They have characteristics of both white fat and brown fat, and potentially function of both white fat and brown fat. And so really understanding beige fat is kind of a newly discovered type of fat in the last maybe 20 years. Trying to understand exactly its role is really hot topic. So where can you find these fat depots? So white fat, as you might all be aware, is kind of everywhere. There's always a layer of fat underneath all of your skin. That's your subcutaneous fat. But then brown fat is actually found, it's called interscapular brown fat. It's right behind the back of your neck. For many, many years, it was thought you only had brown fat when you're first born. So brown fat's function seemed to be when you're first born, you have no ability to regulate your temperature. So your brown fat actually has the ability to do what's called non-shivering thermogenesis. So it can uncouple the electron transport chain and essentially produce heat instead of energy. And that's how newborns regulate their temperature when they're first born. And it was thought as you grow, your brown fat shrinks and eventually disappears because there's no need for it. Well, it turns out that does not happen. We still all have brown fat, and it's predominantly right behind our neck. And white fat, again, can be sub-Q, which is that layer kind of under your skin, and then there's visceral fat, which is more of your abdominal kind of fat. And so these are the depots we study in trying to understand what might be going on. So what I did was I wanted to generate a mouse model. So what happens if I made a mouse that could not express ACLY in its fat? So I should tell you, if I delete ACL from the whole body, so if no cells in the body can make it, it's an embryonic lethal, meaning that these mice die in utero, so they're never born, suggesting ACL is a very important protein in development. It's a whole new function for ACL we were not expecting. And I don't know if anybody's actually studying it. You would think we should study that, because that sounds really important. But it has not yet happened. So, since I had no mice, I decided to make a conditional knockout, meaning I only deleted ACL from the fat. So, I made these mice. And what I wanted to do was study what effects this would have on the mouse as a whole body. So I wanted to look at their weight, their blood glucose levels, their insulin tolerance ability, glucose tolerance, as well as look at their tissue. So I studied these mice over a 16-week period, and I found that over 16 weeks, my ACL specific knockout compared to their control litter mates had no difference in weight. They all seemed to gain weight at a normal rate, so I thought. So then I wanted to look at their ability to use glucose and insulin. So glucose tolerance measures the body's ability to respond to insulin. So your body, after you eat, your body will get a signal in the pancreas to produce insulin. It will secrete this insulin, and that's a signal to all the cells, mostly your muscle and your fat. They're the main workers in this case. That insulin tells them to take up the glucose out of the body and either use it as energy or store it as fat. And so we can do these glucose tolerance tests to see if the cells are able to take up that glucose. We can also do an insulin tolerance test where we can see if the body is even able to respond to that signal to begin with. And this is just to remind everyone, so we produce insulin after we eat, and then they will tell the body to take up glucose. The brain actually does take up some, but the biggest users of insulin or responders to insulin are our muscle, fat, and liver students. So first I just wanted to see what their normal glucose levels are. So I fasted them for 6 hours and just looked at their normal fasting blood glucose. What are they at, like a basal normal level? And I actually didn't see any significant differences. So they seemingly, my knockout mice versus my wild type mice had no fasting blood glucose level differences. So then I decided to do my tolerance tests. And this is just an experimental outline for how I set it up. So I actually fast the mice overnight for about 18 hours. And then after that, I come in and if I'm doing a glucose tolerance test, I'll give them an injection of glucose. And for the next 2 hours, I will monitor their blood glucose levels every 15 minutes. If I do an insulin tolerance test, they get an 8-hour fast, followed by an injection of insulin, and then monitoring of their insulin levels, or glucose levels, sorry, over 2 hours. So I did the glucose tolerance, and what you can see is this is time 0, so they all had pretty similar fasting blood glucose levels. I administer the shot of glucose, so that's going to raise their glucose really high, so you can see this really nice increase in glucose. And over the next 2 hours, you can see it coming back down to normal levels. And what you might be able to appreciate here is that there was really no significant difference in glucose tolerance between the two animals. So we did my insulin tolerance test. Again, here's time 0. They all have a similar level of glucose at this point, blood glucose. I administer a shot of insulin. So I told you insulin tells the body to take glucose up. So after I give the shot of insulin, you can see this really dramatic decrease in blood glucose. And over the next 2 hours, it comes back up to basal levels or normal levels. And again, I actually didn't see any significant difference. So at this point, I was like, this is weird. But I was like, let's keep going. Gotta figure this out, right? So then I sacrificed the mice and looked at their tissue. So I started by looking at their white fat. And what you can see here is this is my wild type or normal animals, and these are my knockout. very similar. I didn't see any difference. I also looked for potential inflammation. I don't have to show you because it was not there. So again, I was kind of disappointed because these mice aren't doing anything. But then I looked at a different white fat depot, their visceral fat, which is kind of like the abdominal fat. And what I found was the knockout animals had larger adipocytes. And this is usually a characteristic you see in obesity or other metabolic dysfunction, sometimes diabetes or non-alcoholic fatty liver disease, things like that. So I thought that was interesting. So this may be a primer, or maybe the mice need to live a little longer for you to see disease. That was interesting. But what was probably the most interesting was when I looked at their brown fat. So on the left, here's my wild type animal. This is what normal brown fat looks like, and this is my knockout animal. This looks like white fat. So I'm having my knockout animals are making, or their brown fat is whitening. This is very rare. There are almost no conditions where we've ever seen this happen. So brown fat is a really hot topic because once we rediscovered that humans have brown fat after their newborns and continues to live there, we found brown fat's job, as I told you, was to make heat or use glucose to make heat instead of ATP or energy. And it turns out that process uses a lot of energy. So if you activate brown fat, you actually lose weight. So people became really interested in brown fat in the last 10 years, trying to find ways to treat obesity by activating brown fat. So one of their ideas was, well, one of the things that triggers brown fat to be active is cold. They found people who live in Scandinavian countries are more metabolically healthy and not, they don't have the obesity problem we have in other countries. So unfortunately, I don't know how they got approval to do this. They put people in the cold for extreme amounts of time. People die from that, so it didn't work. Obviously, I don't know, but they still had to try it. But they're still trying to find ways to make your brown fat become active and expand, because in mice we can see that this has really great effects. So one of the other things they try to do is take some of your brown fat and transplant it in your white fat, like if in an obese patient. And what we're finding is sometimes actually the brown fat can kind of take hold, start to grow, and affect the metabolism of your white fat, leading to weight loss and improved metabolic health. So in my case, I had the opposite effect. Instead of improved brown fat, I have whitening, which is bad. You don't want to see that happen, right? But, and I should also show down here, so when I take the mice and I sacrifice them, I remove their brown fat, I remove all their fat depots and take pictures. And this is a picture of their brown fat depot. So again, in a mouse, it comes from the back of the neck as well. This is my knockout mouse, and this is my wild type. The size, I don't know yet if it means anything. I've not been able to figure that out. But I don't know if you guys can see it, but the knockout one is actually whiter than the wild type. So just to show that not only do I see this at the histology level, but the whole depot is whiter. So this just suggests that there's some sort of lipid or fat accumulation happening in the brown fat that should not be happening. And this is caused by ACL not being present. So as I said, brown fat has the capacity to burn glucose and fat to reduce heat, and it's really activated upon cold exposure, and its activation is really associated with reduction in obesity. So I kind of just wanted to take a step further and look at the actual gene expression in these tissues just to make sure I'm not missing something. So I looked at my white fat gene expression, and these are just, this is ACLY, the enzyme of my interest, so you can see that it's pretty much not being expressed in my knockouts, which is what we would expect. Then I saw this ACSS2, which was, if you remember at the beginning of my talk, the only other known enzyme to be able to make acetyl-CoA in the cell. It is dramatically up, which may be the reason why I actually even see fat production. Because when we knocked this protein out, we were not even sure that these mice would be able to make fat. They clearly make fat no problem. So maybe it's this protein here that's compensating. Now, up until this point, no one's ever been able to show that this protein can compensate for ACLY, but clearly it's dramatically upregulated. So it's something we're very interested in trying to understand. But what I also found was these other genes, FASN, glucophore, and KLB, which are just genes that are necessary for the adipogenic profile, meaning in order for a fat cell to be completely made, these genes need to be used. And they're all kind of down, although fat is still being made, suggesting that even though the fat gets made, there might be some metabolic defects in these fat samples. I looked at some other markers. These are just markers of adipogenesis again, and they're all down, suggesting a similar effect that even though the fat gets made, there may be metabolic defects. So then I went on to look at the brown fat depot and look at the genes that are being expressed there. And again, I saw this ACSS2 dramatically upregulated. And I saw some reduction in this glucose transporter, suggesting they're not able to take up as much glucose as they might need. But what was most interesting, where'd it go, is down here. So all fat express markers specific to that depot. So there's white fat, visceral fat, specific markers, brown fat specific markers. So when I looked at the brown fat specific markers, they were all down, suggesting they're becoming more white. I tried to look at some of the white markers in these brown fat, and actually it was, one was up, one was down, so I can't really say that they're becoming white fat necessarily, but certainly the brown fat markers were all down. So at this point, I was like, all right, I have my mouse data. It's not really clear that what's going on. There was no major defects in their insulin tolerance or their glucose tolerance. Their weight gain was completely normal. I had this really interesting brown fat effect that was not really clear why I was getting. So I was like, well, can I recapitulate this system in vitro or in plastic? So without using the mouse model. So what I did was I took fat from these mice, I digested it, and isolated the cells. And I immortalized them, meaning I was able to make them proliferate and grow consistently in response to the stimulus. And then I administered a virus, and I was able to knock out ACL in my cells. So I was able to make a cell line that recapitulated what, or I hoped, recapitulated what I was seeing in the mouse. So maybe I could try and study this in another way to try and understand what might be going on. So what I first did was, well, are these cells really doing what I was seeing in the mouse? So let's look at insulin. So I basically, so I've already told you a little bit about the, if you're impaired for being able to respond to insulin, you're what we call insulin resistant, so you're not going to take up the glucose you need. And insulin signaling is actually a very well studied process. So when insulin is secreted in your blood, it binds to something called the insulin receptor, setting off this like whole cascade of signaling. So we can actually probe for these different parts of the signaling process to see if insulin signaling is happening the way it should be. So basically, I looked at, So I looked at all the parts of the insulin signaling pathway, and I found, I did this by Western blot, which is a way to look at protein expression. So what you see here is, first I looked at ACL, which is kind of my control to make sure my cells are not expressing. So here's my wild type cells all expressing ACL as they should. And here, where I knocked out, you don't see any bands, suggesting there's no protein being made. So that's a good start. I'm not making ACL. Then I looked at these different aspects of the signaling, insulin signaling pathway, which are known as AKT and S6. What you hopefully appreciate is, so what I do is I do insulin stimulation to the cells, similar to what I did with the mice, but instead I do a few different concentrations of insulin just to see what kind of response they get. So a low concentration all the way up to high. And in my normal wild type cells, you can kind of see this like nice increase in insulin signaling with increasing insulin concentration. So that's really blunted in my wild knockout cells, suggesting that these cells are resistant to insulin, which is not what I saw in my mice. So I also wasn't able to upload the original talk. I had some other data, I'll just summarize for you. So one other really cool thing we can do is called metabolic tracing experiments. So we can actually take glucose and put a label on it so we can follow its path in the body of the mouse or in the cells. So what this can allow us to do is see where the metabolite might be stuck, so we can get an idea of what metabolic pathways might be affected by our mutation, by deleting ACL. So what I did is I took a glucose that had this label on it, so I was able to label all the carbons in glucose with a label, and I injected it, well, first into my cells, and over time, I was able to then look at where that label went. And what I was able to see is that my normal wild-type cells, glucose gets taken up, and it gets used just as we expect, so it goes through glycolysis, it gets put through the TCA cycle, and then used to make fat. And we can see really nice enrichment into fat. My knockout cells, when I do the same experiment, are not able to use the glucose. So either it's not getting taken up, or it's getting stuck somewhere in the glycolysis TCA cycle pathway. So then I followed that up with another experiment where I actually tested the uptake. I was actually able to see that my knockout cells are not taking up glucose, suggesting that there's an impairment in their ability to, since that glucose transporter is not getting expressed at a high enough level, it can't take up enough glucose to generate fat. So at this point, I'm actually very actively pursuing this brown fat phenotype that we're very interested in. And what I'm doing is whole body metabolite tracing experiments. So I have a glucose that's labeled and I have acetate that's labeled. Because I'm wondering, since I see this ACSS2 protein up, if my cells are actually taking up acetate instead of glucose to try and make fat. So I'm in the process with the radiology department to use their PET scanner to see, so we can actually see where the acetate goes because it's labeled on a whole body basis. So I'm really excited to get that data, and that's probably the biggest thing I've been working on. The other thing is, I showed you this brown fat looks very white. So I'm wondering if this brown fat can even function the way it is, or if it's a developmental problem. So is this brown fat never getting made right? Or is it getting made and then there's some effect later on as the mouse ages? So I'm doing cold studies, so you can regulate temperature of the environment the mouse is at to see if we can activate the brown fat. And we're also in the process of doing those studies. Fortunately, I don't have that data yet. That's kind of like the ongoing experiments we have. So yeah, and we're very interested in taking my mice. So initially, again, I didn't see any real metabolic defects, right? But what happens if we subject them to a high-fat diet or a high-sugar diet? So my protein ACLY has actually been shown to be really important in suppressed by a high-sugar diet, which is what we call a Western diet. So in Western countries, the U.S., we consume a ton of carbohydrates and sugar. So we call it a Western diet. So I'm going to subject my mice to a Western diet and see if I maybe exacerbate any of these effects. Yes. So I just want to thank a lot of people who were extremely helpful. Mostly my amazing advisor. She is probably the most patient and enthusiastic human being I've ever met, and I'm grateful to be in her lab. And all my lab mates, who I have to tell you, are just awesome. They love talking science. And we have just a really great work environment. Really lucky. Some data I really didn't get to share with you, but that is equally exciting in my opinion, at least. I do in collaboration with Nick Snyder and Tony Mancuso. So I just want to thank them, even though I didn't get to share with you that way. And I would be happy to take any questions.
Could you repeat the question?
Sure. I think she wants to know about my background as far as. Oh, okay. Yeah. So we were interested. So this protein ACLY has been shown in human patients with obesity and diabetes to be suppressed, and we didn't really know why. So basically, we had some preliminary data suggesting that when you suppress ACLY in mouse models or in cells, that we see effects on glucose transport. So we hypothesized that ACL might be playing a really important role in normal adipocyte function or fat cell function. So we were interested in seeing if we could get to that root of that problem by making a mouse model, because no one's ever studied ACL specifically in a mouse model like this. So, does that answer?
Are there any other questions? Please take advantage. We have another 7 minutes.
Why do we, as an audience, need to know this?
Oh, that's a great question.
Why do you, as an audience, need to know this? What's the significance of her research for your life?
Yeah, that's a really good point. Maybe that question is something I should put more into my talk. But so, as I mentioned early on, ACL has been very, at this point, it's being very well studied in the cancer field because we see it highly expressed in many cancers. And because of that, there are now 3 drugs in clinical trials that target ACL. However, people seem to ignore ACL's main function, which is in lipid synthesis. And so why you should care is if any of these drugs get past clinical trials, this work is really important in showing side effects that those drugs can possibly have. And I think a lot of people, you go to your doctors and you kind of trust them that they're going to put you on the right drug, make the right decision, but I think it's really important for people to understand, all the other effects that these things can have. So I showed you this really dramatic effect on brown fat. And if your brown fat is not active, you're not able to burn glucose the right way, you're going to have some maybe overall metabolic defects. And if you're on a drug that's going to cause that, I think we should know that.
What about the weight loss fat drugs that target your metabolism? Do they affect your brown fat, or is it just specifically white?
Wow, so yeah, we're finding more and more that they do affect brown fat. So initially, a lot of these drugs, nobody studied brown fat for a really long time. So some of the more common drugs, weight loss drugs, as well as actually anti-diabetic drugs, so some of the drugs people get on when they have diabetes, are what we call targets of master regulators. So they affect proteins that are required in this making fat process, or in the upregulation of certain aspects of these fats. So yeah, some of them actually have negative effects on brown fat, they're finding. It's still a really active area of research. It's not completely clear, but so what we're finding is a drug that may work really well on white fat has the opposite effect on brown fat. Yeah, that's a great question.
And another question is, do you see yourself, you and your team, in the future applying these types of drugs that they use for givers onto the small mice, or is it just strains of them?
So some of the drug companies that do some of these clinical trials have given us some drugs yet to test. We're not ready yet to start administering drugs to our mice because we still need to really understand exactly what's happening first. But that will definitely be something we'll do a little further down the road. We have, I think, 3 different drugs that are not yet some of them are not even in clinical trials, some are just in clinical trials. They've asked us to test them on mouse models. We're not ready.
So the drug companies go to universities like yours and whatnot to...
Not always.
Not always?
No, it depends on the drug company. Our lab, so my boss is probably one of the founders of this protein, ACLY, so she is considered kind of like the expert in the field. So because she kind of knows a lot about it, some drug companies have come to her for these purposes. A lot of the big drug companies will have their own labs where they do this kind of stuff. The smaller ones where they're maybe not set up to do these kind of experiments or don't want to invest in setting it up will give us. Or in one case, actually, there was a drug company that's now been sold 5 different times. So I don't even know what it's called at this point, but it's a small one that had a drug that they thought would be important or helpful in Alzheimer's disease. And it failed clinical trials, but and most drug companies at that point just throw the drug away and continue on. Some scientists that was part of this trial noticed in some of the experiments they did that it was binding to ACLY. And he's like, well, this could be something really interesting, maybe not in this context, but in another. So they sent us the drugs to try and figure it out. We've not yet figured it out, by the way. But yeah, so every once in a while something like that happens. I don't know how common it is. I think it depends on the lab.
Any final questions? We're running out of time that you'd like to know about fat's role in the body, both beneficial and detrimental to our health.
You had mentioned something about inflammation
Yeah.
and the effects, what affects that.
Yeah, so right now the whole inflammation story is kind of a chicken and egg story. We're not sure which causes which. But we do know we see with ACL knockout, we see increase in some inflammatory markers. We also see increases in something called IL-6, which for those of you maybe in the immunology field, has always been considered to be a pro-inflammatory, it can be anti-inflammatory, depending on its context. And there's some recent data suggesting actually IL-6, when it's secreted from our brown fat, is really beneficial in helping brown fat be active. Excuse me. So we're seeing some IL-6 as well. But we don't know if the inflammation is because we deleted ACL or if the inflammation is kind of already present. Yeah, something we're very interested in trying to.
Any final question? All right, then let's thank Dr.